Okay, I'm going to take a slight detour before I start on today's lecture, because I'm guessing that I'm probably not going to get through everything, because I realize that we've had these mentions of these techniques, and I think it's kind of important that we understand what they are. And I think, unfortunately, many people, including myself, don't fully understand them. So I want to talk about three in particular, the last one not so much, but the first two in some detail. So first one is FRAP. Anyone want to tell me what FRAP stands for? Yeah, I'm not going to write it out because I'm lazy. So fluorescence recovery after photo bleaching. So you have a system in your field of view. You have let's say, a fluorophore tagged, let's say just everywhere in your system, but you basically use a high-intensity laser to effectively bleach a specific region. So this is photobleaching. If you overstimulate a fluorescent molecule, it basically goes into its triplet state, and essentially we can think of this as a dark state, where it doesn't fluoresce, and this is quite a long-lived state, and so essentially your total intensity goes down. So the idea is that you bleach all the fluorescent molecules in a specific area. So this area goes dark, and the outside stays light. And then what you do is you record what the signal looks like in this area. Because what will happen is the molecules around will start moving inwards. And so your signal was here, you bleach, so your signal goes down, and I'm only looking at the signal inside the bleached area. And then as molecules from outside, for example, move in, your signal recovers, all right? And this is time, this is signal. And so from how fast this signal recovers, you can try to infer some dynamics. Now, there are two important things you can generally do. The standard thing is to fit this function to something like, right? So it's often an exponential-like function, and this parameter alpha, which is, okay, actually I'm going to define it differently. Sorry if you've taken notes, but I want to do it differently. This parameter tau, which is a time scale. So this tau is defining the time scale basically for how long it takes the signal to kind of recover, all right? And then there's a second parameter which people often focus on, which is the difference between the before signal and the final steady off signal. The recovered signal. So what should I call this? Just call this ID for the difference in intensity, right? And if you go for literature, people try to use this data to try and infer things. Now, everything up to this point is kind of standard. You just take your system, you bleach it, you make a recovery time, you get this time scale tau. What happens next is where problems arise. Sorry, Vijay.
The picture is that the molecules are coming in, and that's why there's no turnover. It's not like
a membrane. I was going to get to that, but yes. So the important point is that the tau is the recovery time, all right? So that's how long it takes for the system to recover. The problem then comes with what you do with this. So Vijay has basically pointed out, to interpret this tau into a meaningful biological parameter, you need a model, all right? This tau in of itself is not meaningful. It is very meaningful, sorry, but it's not like, you cannot say this is the diffusion time or this is the turnover time, all right? You need a specific model for what you think is going on. So as Vijay said, what you can have is inside your volume, you can have turnover. So you can have new protein produced. You can have new folding of fluorophore. That would give you a recovery in the signal without any movement from outside inwards, all right? In that case, this tau is measuring the turnover time. It could be that you have lots of molecule outside and you're looking at them moving into that region, in which case this tau, so this tau could be related to turnover, and it could be related to transport, right? Of which the most obvious one would be diffusion. And so the key point here is that you have a time scale, but that time scale doesn't necessarily mean something that you want it to mean. You have to apply a model to it to interpret that time scale into something. And this is where a lot of problems come, because people just naively put in their model, i.e. diffusion, and they then infer diffusion constants where that's not necessarily the correct thing to do. There are also other major issues with FRAP. A very common one is the following, that always look carefully at people's y-axes, because you'll see often a thing like 1, 0.95. What's gone on? This is actually the honest version. This is how you should do it if this happens. Why would you have this? Whereas the theoretically perfect FRAP is, this should be 0. Like you make your region dark. Why isn't it like that in a real experiment? Background is 1, yes?
We don't use very strong labels.
Not quite, no. You could bleach fully. This is assume that the system has correctly done it. Anything else? So often there is a time lag between when you actually bleach and when you start recording. Most microscopes have them on different setups, or they actually use the same laser and then they try to switch between the two. So in reality, the system did this down to 0, but you don't start measuring until the signal has recovered to here. So in fact, you see a very small change in the signal. And so now you're trying to fit a curve with an exponential over sort of 5% of the curve, all right? And so your fitting can therefore be, can be rather dubious. So essentially what's happening in this case is that you've bleached your area with and in the time that it took you to switch your microscope from bleaching to recording, your system's already recovered some of the signal, right? So this is the bleach. Let's call it crossover. And then you've got recording, right? And now you're seeing the final bit of the recovery of the signal. And so this is a very common mistake. If people show it like this, is actually fair. This is what they saw. It's actually perfectly good. What you should be very careful of, and why you should always read methods, is when people subtract off the value at the start of their recording to define that down to 0, right? Because that's really dodgy. And people do this, and it's because they don't understand what the measurement means. So always go through people's methods when they talk about FRAP to understand whether they're actually showing you the intensities that they should be showing you. But that's just a technical issue, and but still you recover a timescale. Then also with FRAP, you have this fraction of unrecovered signal. Now, can anyone give me what they think may be causing that difference? I actually had one answer already. Yep, so, but what do you mean by binding? But binding is the correct answer, but in a specific way. So I just wondering if you could give it a little.
If you have a protein which binds
to
another, so diffusing proteins would bind, and when you teach them, they would be there.
Yeah.
So it can't be replaced.
Yeah, exactly. So you have basically what we call sometimes it's called a dark fraction. So these are bound molecules that stay bound to specific sites. So when the new molecules come in, they can't go there because they're already populated. And so you have essentially what's called the bound fraction. What's another cause for this difference? A technical difference, not a biological one? No? Yep, so photo bleaching. Again, always go to people's methods. How do they correct for photo bleaching in their FRAP? I can assure you that most of the time it's not done very well. And particularly photo bleaching, you should work out by doing a control, where you do it with the same fluorophore under the same imaging conditions, but without whatever is being bound or the protein of interest, and actually looking at the recovery of that signal in a pure sense. And then you look at it in your applied system. People often try to correct for photo bleaching in ways which I find somewhat dubious. and then you get a bound fraction. So this difference is always this combination, and unless you're very careful, it's sometimes hard to distinguish between the two. Yeah, so this is the next good point. So typically, you can do FRAP experiments on tiny regions, but it would be stupid. Typically, people, I guess, do them on around 10 microns, 1 to 10 microns. Munia, what size is your FRAP experiment on?
I'm trying to look at them supplementary.
But we've done it in our lab typically on 5 to 10 microns, a single nucleus often. I've done it on fission yeast cells, so 1 micron. So that's typically the scale. And the important thing here is what time scale are we on? Typically, we're on the order of minutes. Seconds to minutes, okay? So you're looking at the recovery of the system as a whole. Now, this means that often people like FRAP, and there are quite a few reviews where people say, oh, FRAP, and I'll get to FCS in a second, but FRAP is better than FCS because FRAP is measuring the relevant time scale. Now, I think that's questionable what that means, but what they essentially like to think is that, well, the processes I'm interested in, like gene regulations on, are happening on the scales of seconds and minutes and so on. So therefore, because the FRAP is measuring on those scales, it's the correct measure. I think that's a tad naive, but I think so. But that's the argument for why it's there. So what you're actually seeing, and I had a good chat with Frank before giving this talk, is that this TOR and the subsequent parameters you measure are really effective parameters, all right? They're measuring the cumulative effects of diffusion back in, binding, degradation of your molecule. You can imagine other protein-protein interactions. These are all embedded within this time scale, all right? And so it is, I think, whenever you have a parameter from a FRAP experiment, I think it should always, unless you can really prove it, be preceded with the word effective. Our effective diffusion is ... Our effective bound fraction is ... Because it is a measure of the cumulative effect of multiple processes on the system. It is too slow a time scale to be measuring the actual interactions. Okay, because as we just heard from Munia, all the actual interactions, the DNA binding, the diffusion, these are all happening on millisecond, second timescales. So FRAP is too slow to measure the actual sort of on-the-spot dynamics that are happening. It is measuring the effective cumulative effect of those interactions on the system. Would you say that's fair? Okay. There's also other issues with FRAP. You're blasting this area with high laser and heat. People have actually shown that you damage this area. If you're doing, for example, in my field, mechanobiology, looking at actin, you're burning your actin, you're burning your cytoskeletal structures. That actually changes their property and how it recovers. And so, again, when you do FRAP, you really should actually do it with a range of powers and actually check your timescales are consistent, right? Because it could well have effects on the physical structure that you're trying to measure. Essentially, it's like mild laser ablation, all right? And so that's also another critique that you need to be aware of when it comes to FRAP. Okay. Any questions about FRAP? Okay, so what does FCS stand for? You know from Munia, she said it in her talk. Yeah. Again, I'm too lazy, I'm not going to write that out. So how does FCS work? This is my artistic impression of an objective. And our objective creates a light cone, and we have our sample here. Now, there's an important point to realize here. Does anyone here not know what a point spread function is? Everyone? No one wants to put their hand up? Okay, I've got one or two. Okay, just as an important point, like, as a physicist should know, you, of course, when you image something, you are diffraction limited. This is why we have to get cheats around. They have things like super resolution. But in a normal microscope, your, oh God, shouldn't do this off the top of my head. 1 point. Is that right? This is the Okay, that's lucky. So essentially, the smallest size that you can measure using a light with a wavelength of lambda is given by this, and then Na is your sort of the numerical aperture of your objective, how much light you can actually observe. And so this typically is around 250 nanometers. So this is saying that if you have two molecules closer than 250 nanometers, you cannot distinguish them. They look like one blob, okay? I'm not going to go into how you can get around that, but you can. And unfortunately, in this type of in normal microscope, so this is this delta X here. You also have a resolution in, and I always get these dimensions the wrong way around. You've got axial and call this delta Y. You're basically your, I want to say axial, but I think that's the wrong word. What's this dimension always called in microscopy? Lateral, thank you. So the lateral resolution is actually bigger, and I do not want to go into why, but typically it's around 1 micron in a confocal. So what this is saying is if you have your perfect light microscope, you focus it into a perfect spot. At that spot where you've got focused it, you've actually not got a spot, you've got an ellipsoid of roughly 200 nanometers by 1,000 nanometers. You can get this a bit smaller and so on, but that's the basic idea. So what does FCS do? What FCS does is that you focus on this one point, which is in fact an ellipsoid, and you just measure the intensity in that point. Typically, you just have a photon detector, so you don't have a camera on a good FCS machine, because cameras are slow. You just have a detector that detects photons. So there's no spatial here, you just have a single spot. And what happens? Well, let's say let's use a different color. Where's my blue gone? Oh well. Let's say I have 4 molecules in my spot at this point. So I'm going along, this one diffuses out. My intensity goes down. This molecule then comes in, so my intensity goes up. Another molecule comes in, intensity goes up, this one leaves. And so you end up getting an intensity profile that looks something like this. And you may think, well, this is pretty useless. What the hell do I do with this? And what you can do is interpret this into what's called the autocorrelation function. And that is essentially, I should have looked up the actual definition before I came. I'm going to get this wrong. Essentially, though, you ask, what is there's some denominator as well, but essentially you just ask, how similar is the value of my intensity sometime tau later than it was at the time I am now? So basically what I'm saying is that when tau is very small, my signals are highly correlated because you're essentially asking in the very next little second how fast this is. And in an FCS machine, just so you know, this is on the order of 10 to the minus 6. We measure typically 10 to the minus 6 to 10 to the 0. So totally different time scale. You're going now from microsecond to second versus FRAP, which is second to minute. And so if you look at 2 extremely close together time points, they generally are highly correlated. So you have high correlation. And then as you go further and further away, this point with the next distance one, they get more and more uncorrelated. And over time, your signal goes down to 0, right? Yeah.
T is a function of tau. And
yes, sorry, this should be tau, yeah. And this is an average. And you average over many, right? So now we can ask, well, let's just use some intuition. Let's not worry about getting the formula for G in terms of a diffusion. In fact, I used to know how to do it, but I couldn't remember, and I didn't have time to look it up. But you can still get some intuition. Now, let's compare. Let's imagine we had 3 profiles like this. In this one, the correlation goes down very fast. What that means is that the molecules in this volume are rapidly changing because they're not related to each other. So this would imply, in this case, that D, the diffusion constant, okay, actually, no, I don't want to do this. I don't want to make this mistake. This implies that the time scale, using Tor, I want to use Tor for time scale, so I'm going to change this to capital T. The time scale over which the fluctuations are correlated is small. And a good example is if they've got very fast diffusion. Because if very fast diffusion, they'll move out rapidly, and so you get basically extremely rapid fluctuations, and so the correlations are small. If they move extremely slowly, even to the extent where they're almost bound, right? So they're there for a long time, then so this is T fast. Then the time scales are much longer. The molecules are around each other for longer, so the intensity profile stays at similar levels for longer, and so therefore you get a much higher autocorrelation, right? And so you can, again, from these experiments, you can get time scales for the dynamics of your systems. Now, these are time scales on the order of microseconds to seconds, okay? And so that means that, for example, as Muni was alluding to, you can see binding events, all right? Because binding events are happening on the order of milliseconds to seconds, which you cannot see in FRAP. Now, again, though, so what we are measuring in FCS are these timescales. To convert those timescales to something that is useful, or not useful, but something you think is more meaningful, again, you have to apply a model. All right? So your model could be, the classic one is diffusion, right? And in a simple diffusion case, you can get remarkably good fits to this. And from that, then you can convert your timescale into a diffusion constant, right? But again, that is dependent on the model that you choose. And this is something that people often overlook. They just apply a diffusion model without much thought. Because this is sort of on the order of, because actually, yes, I should also have made this clear. This is a log scale. So on a log scale, basically on really short time scales, so 1 microsecond, you basically are the same. Like, if you, two molecules are together, for them to have left the volume inside 1 microsecond, the probability of that is very low. So the correlation stays high. It's only over long enough timescales is it likely that one of them will have left. And so that correlation will have changed. Well, because, so in these short timescales, basically what it's saying is, on these short enough timescales, not much is changing. So the correlation stays high. And then as you get to long enough timescales where things start to change, i.e. binding events, new molecules coming in, molecules leaving, then you start to see decrease in the correlation. Nope. So that was the next point to get to. So in fact, when you fit this, you can get 1 timescale, but often actually, and traditionally the old FCS, if you go to FCS papers from 10 plus years ago, they always just measured 1 timescale. And often, to be fair to them, they would say, we know that there's probably more, but that's not the case. But with modern FCS, so just I think a good lab, an experimental lab that has done a lot of very beautiful work on this is Nico Plachta's lab, does this in mouse. You can actually really start to get to the point where, let's go out to 10 to the 0. So what you often get is what people now call a diffusive timescale, which is the fast, rapid turnover inside the volume. But you often see a long tail if your FCS is very good. And that long tail is telling you probably more about things like, for example, binding. Because the binding is generally a slower process than diffusion, yeah. Well, so in fact you can. So I guess another, both of them are in Singapore actually. So Torsten Voland is an FCS expert, and so they've actually done this in zebrafish, where you can measure the binding and unbinding rates as well. And so essentially what you can look at is, what you interpret is actually two timescales. You get like a T1 and a TOR2. And again, you then have to apply a model to interpret that into something. We, in fact, in the lab now have extremely nice FCS data on bicoid. We've been working with Torsten now for a while. And bicoid has two timescales, both in the nucleus and in the cytoplasm. So to come back to a question I was asked yesterday, which I realized I didn't answer, which is, what's the diffusion of bicoid, right? Well, the diffusion of bicoid is typically around 5 to 10 microns squared per second in the cytoplasm. Now, to give you an idea, free GFP diffuses at 65 microns squared per second. So that tells you that the bicoid is not freely diffusing. In fact, it's probably interacting with actin and the cytoskeletal domains, and that actually slows it down. And we can see that in our FCS. We actually see very rapid, small level diffusion, and then also a longer time scale, which is probably interactions with the cytoskeleton. And then in the nucleus, we can also see two time scales. And there you're probably seeing binding on the transcription and diffusion within the nucleus. The one point I want to make about FCS, which is kind of cool about it, is FCS is a rare technique where you want to have low numbers. Why is that? Because in FCS, you want to measure fluctuations. So if I had 1 million molecules, and I'm looking at, oh, one left of volume. Wow, I have 999,999. Like, my fluctuations are small, right? If I have 10 molecules and one leaves the volume, I have 9, right? So I've now got a 10% change. And so it turns out, in fact, that the value, I put this to 1, but that's not necessarily true. The value of G of 0 scales as 1 over your particle number. So you want the smaller the number, the better. Because if your n is because basically if your n is too big, what you see is this. And you will actually see papers where they then try to normalize this and fit it, and it's all not very pretty. Again, when you see FCS curves, always check their methods, always check how they normalized the data to do the fitting. Whereas if you've got low n, then you tend to see this. So what Nico Plachta realized is that he used photoactivatable GFP. So now what he used was just very, very low levels of ambient light to activate just a few molecules. And so now when he did his FCS curves, he got beautiful, his lab got a beautiful FCS curves because they had low number. One thing that we realized, we tried doing that in Drosophila and it failed. For some reason, PA-GFP in the early embryo is a bit funny and clusters. But of course, what's nice is that bicoid is a morphogen gradient. And so there's lots of it here, but of course, as you go down, it gets reduced. So if we plot the profile, bicoid looks like this. Now, people who did FCS before in bicoid did it here. But that's not the best place to do it. So we've been in the lab now, we do our FCS here at around 70% along the embryo around this point. And we get much, much better FCS curves. And so generally, when you're doing FCS, what you want to be is you want to have as few molecules as possible so you're above the noise. Because obviously at one point you go so low that you're just actually measuring background and shot noise on your camera and some ambient noise that you get from that the room's not completely black, right? But so you need to be above that background level, but essentially you just want to be above that background level.
In terms of concentration, so what would be for you the kind of minimal concentration? What's the range of concentration for which the
I'm trying to remember. So Thomas claimed that at 50% into the embryo, there was around, bicoid was about 6 nanomolar, I think. So I guess we're measuring it around 1 to 2 nanomolar, I guess, so very low concentrations.
The other point was that right, you did that so that you have bicoid hubs in the nucleus. So haven't you tried to within the hub?
It would be in principle. I guess the problem with the normal FCS setups don't really have live imaging as such on them. So it's hard to identify specific points. You can, There is a technique called number counting, which is related to FCS, where you actually try to look at the change in the steps. You try to infer this as step jumps rather than just continuous. And then you say, okay, that was maybe two molecules binding or coming together or something. And so I've never been very happy with it. So there are techniques, but FCS itself doesn't directly do that. And it's still a model, right? You still have to apply a model to get these numbers out. But I think the key things really is FCS generally you can get multiple timescales. FRAP you generally don't. You get timescales which are on the order of the actual dynamics, which FRAP isn't. But of course, what you're seeing is it's a little bit, you're looking at the trees. The argument would almost be that FRAP tells you about the forest and FCS is telling you about the trees, right? And the point is that really neither is correct. To get a good view of a system, you should know both, all right? And I think that's generally, my feeling is that you get the die-hard frappers and you get the die-hard FCSs, and they kind of are like, guys, both are good systems, and you should use them sensibly and know the limitations, and I think that's often the problem. Where FCS is bad is if you do not really, really, really know what you're doing, you're going to be measuring noise. So I actually work with an FCS lab because I'm nervous about FCS, because you're measuring noise, right? That's the whole point. But are you measuring real noise or are you measuring your incompetence noise? And I wouldn't trust myself on the microscope. But so we work with a FCS lab to really control this. And a big, big issue in FCS is photobleaching, because you've got very low numbers. Your bleaching makes a real difference. And so you really must control photobleaching effects. So what that means in reality is you must repeat your experiments with multiple laser powers. And essentially what you should find is a plateau where you get the same numbers, where basically what you should aim for if you go laser power, and this is, let's say just one time scale, right? What you might see is something that looks a bit like, this is the ideal case. If your laser power is too low, you're not measuring anything. You're measuring noise or background, and so basically you get some random number. You get into a regime where your laser power is high enough to image and get readout, but low enough that you're not bleaching significantly, right? So this is where you want to be. And then you get into a regime where the time decreases rapidly. Why is that? Because if you bleach, then of course, well, okay, this molecule, instead of moving out of the volume, it just bleached. But I lost that signal. So by the autocorrelation function, it says, well, it's gone down quicker. So bleaching does this. So generally, your timescales get shorter. And so you tend to get this sort of profile. And so if you do FCS properly, any good FCS paper should have this. You should have 5 or 6 laser powers where you really show that you are measuring in the correct regime to actually see the dynamics in a reproducible manner. Okay, question?
One thing I have always asked ourselves, because you do several curves, right? Several FCS experiments. Yeah. Do you fit each of them independently?
Yes.
And then, because I think also this
yeah, so in my lab, and Torsten as well, we agreed, we fit every single individual curve and we extract parameters. And then what our data looks like, this is, I could have a slide and show you, but our data looks like this. If I just plotted experiments, right? And you get massive outliers occasionally. Again, you're measuring fluctuations. If someone banged the table during the measurement, you're going to get something weird go on. So what we do is we measure each individual one, we get a parameter set distribution, and then we apply statistical tools to say, well, can we say this is an outlier or not? And then we remove it. It is wrong to average your FCS curves, and people do this. Yeah. Do not average your FCS curves, yeah.
So from a single curve of this photocorrelation, how do you know how many time scales you want to?
You don't.
Why do you pick 2? Why not 3?
No, you don't. The fitting is basically, yes, okay, no, that's a good question. So, yes, how do you know? The rule of thumb that we do is simple. So you do, first of all, a single one, and you get a fit. Is it good? Like, let's say it's, if your data looks like this, then your fit, oh, those are all white. There's green and blue down here. Exciting. If your fit looks like this, I would stop there. Like, you can't infer more information, right? If your fit, which is more common, looks like this, then you're going to be like, okay, yeah. So then you can try a two-component fit. We, in fact, the data that I'm showing here, we do two components, and we have what's called a. So the GFP molecule, you always see this nice image that the GFP gets excited into an excited state, it then relaxes, emits a photon. It doesn't always do that. It can go up to its excited state, and sometimes it can go here to the triplet state, which then goes down eventually. So this is the photon coming out, which is what you want, but sometimes you go to the triplet, and the triplet state doesn't fluoresce, and so you can account for that. And the good thing here is that this has been measured for GFP. The triplet, the time it takes to go between this state and these states is known pretty well. So this is actually kind of a known parameter which we use. And then, but what we do is we do a single component plus triplet, two component plus triplet, three component plus triplet. And then basically you plot your fitting quality. And what you generally see is that as you increase, you've got one component, 1 plus T, 2 plus T, 3, and so on. Your fit quality, like let's say we're measuring an R squared, it basically often looks like this. And then basically, yes, I could fit with 3, but the difference in fit quality is tiny. And so you can't really infer it. Torsten's actually got some nice code where they take a Bayesian approach and essentially ask, probabilistically, how good is it to add? And that gives you more of a feel for it. But generally, in our experience, 2 plus triplet is, you normally see a distinct improvement on the previous one, but adding an extra 1 isn't there. So it has to be done by hand. Again, any good FCS paper should have in the SI those fits, showing the average fits with the different numbers. And then the reader or the reviewer can make an informed judgment about whether they think it's plausible.
You still need a model. What do you fit for?
Yeah, so you fit, the basic model is actually just these timescales, right? And I don't think you can argue that's the model less, but it's essentially, that the timescales have to decrease, and you essentially fit this to extract those timescales. The key thing is then how you interpret those timescales into biologically understandable numbers, and then you have to apply a model. So whether it's got diffusion, binding, trapping, and so on. And that's where some of the confusion comes. Oh, sorry.
When you say multi-timescale fit, so you just do some summation e to the minus t by tau 1, tau 2, tau 3, or?
Yeah, well, it's a bit more complicated than that. I can't remember the formula off the top of my head for what G of tau is. It's, yeah, not trivial. But yes, basically it is. Essentially you just have there's a standard function, which is the timescale for the recovery of the signal, and then you just try to fit it. So the way that you generally do it is you say for a time window of length capital T, you move it along. And then you can get multiple readings. And of course, the argument is that it's, yeah. Well, you basically, to get this averaging, you just move this window across, and then you record what the value is for each of those windows, and then you take the average of that value. No, I think, okay. Yes, if the dynamics changed, but you're hoping that on the scale of a second or 2 seconds, the fundamental dynamics don't change. You're right. Yes, you could, and if you added a drug or something, you could imagine that. But I've never seen someone test that. That's an interesting idea, but it's such a fast system. I think it would be challenging to do. Okay, any more questions? So in FCS, no, because you're using very low laser powers, because you really want to minimize bleaching, and it's the bizarre thing where you actually want low intensity. So generally, you don't have that issue with FCS. You do have that issue with FRAP, because you heat and damage the area that you are imaging. You basically damage it, and then you look at how it recovers. And so that actually is an issue in FRAP that people often do not properly take into account. And it's often not a problem, but people don't check that it's not a problem. They just assume that the system will recover fine. That's not always true. Yeah. I think if you work out I guess you could work out what the momentum of the photon is, and it's a hell of a lot lower than the general thermodynamic energy scale. So I think you're pretty safe on that. You can, of course, do optic tweezers and manipulate things, but that's at much different energy scales. Optic tweezers are a lot more powerful. But yeah, so that idea is used in biology, but not here.
Is that curve always monotonically diffused?
Yeah, and it makes sense. It has to be, right? Well, I guess, no, does it have to be? No, I guess it doesn't have to be. You could imagine if you had some directed process where you pumped things in and then it reached some steady state and they were fixed in time, then you could imagine it could go up. But I think for any plausible biological process, it is a monotonically decreasing curve, yeah. You'd have to have some active system cheating. And then at that point, what's the point in doing FCS? You've got some active system cheating that go explore that system. But yeah. In any plausible system, I would say that it's going to be monotonically decreasing. Okay, any more questions? Okay, I'm actually going to skip FRET because I have half an hour left. And so I'd like to actually talk about some of the effects. But I think this is important. Like, these are tools that you'll hear a lot. People use them all the time. The really key thing that you actually know, there's one final thing I want to talk about. So when people take this timescale of FRAP, as I say, it's a long time scale, typically seconds, minutes. You get some diffusion constant. And for example, for bicoid, if you do FRAP on bicoid, and you interpret this time scale into a diffusion constant, you get a value of between 0.3 to 1 micron squared per second. If you go and do FCS, I won't talk about the published stuff because I don't believe it, but the stuff from my lab, we now get a fast component, which is around 15, and a slow component, which is around 0.5. Right? Now, the number on the left was the one that was published first. And a lot of people went, whoa, hang on. Your diffusion constant is 0.3. Well, I know what the mean square displacement time is. It's going to be roughly, goes as d times t. So if d is 0.3, and we know from Munier that the early embryo is damn fast, right? You've got essentially 2 hours at most to set everything up. So if we have 2 hours, so 7,200 seconds, that gives us, God, I can't do this off the top of my head, but about 4,000, I guess. Is that right? No. 3,000. Okay. Is that right? No. 2,000 would probably be right. Okay, so that's the mean root squared. So let's now take the square root of this to get the RMS, root mean square distance. So that's about 45? Yeah. I can't draw fives. 45. How long was a Drosophila embryo? You've seen how long was the Drosophila embryo roughly? Not 50 microns. It's half a millimeter. So your embryo is 500 microns. With a diffusion constant of 0.3, after 2 hours, your average displacement is around 45 microns. You can't set up a morphogen gradient. This caused a lot of excitement. A lot of people are like, oh, wow, there must be something else going on. We can have all these active systems and long-range RNA gradients. And then Natalie Destantny published a nice FCS paper where actually they measured around 7.5 microns squared per second. In their case, their FCS curves were not very nice. They were using very old systems. And so they could only do a single component fit. They did the right thing. They actually say in the paper, we think there's more components to fit, but our data isn't good enough, so we're going to play safe. And so they get 7.5. And this has caused no end of problems. Because in the literature, you can find numerous reviews, particularly from people who do not like the idea of diffusion as a viable mechanism for setting up gradients, saying, oh, look, you get FCS gives you 7 and FRAP gives you 0.3, and so therefore it's all rubbish, it can't be diffusion. And they can, I won't name names, but you can go and find the reviews. It's actually slightly embarrassing because it just shows a misunderstanding of what's going on. The FRAP is measuring, I'll answer you in a second, the long time scale of the system, the recovery within a region over the scale of seconds or minutes. This is basically incorporating your nuclear import and export of the bicoid. It's incorporating your bicoid binding to the DNA, the unbinding from DNA. It's incorporating its production. It's incorporating its diffusion, all right? A lot of things. It's the effective diffusion constant in a local regime. This is the movement of bicoid on the scale of a second, how fast it's moving. So the two actually make sense. Why? This is really telling you the time scale for bicoid, the actual movement around is kind of the individual molecule in a sense independent of its environment. It's not independent because it would be 10 times bigger, but it's on the rough order. Whereas the FRAP is telling you about the effective relevant times, the diffusion constant when you include the interactions. And so in fact, how does the bicoid gradient form? Well, it forms through a combination of both transport and the binding. And so you can have, if you actually, then there's a very nice paper, and I can't remember the name of the author. It's in PLOS Comp Biology. It was 2012 or 2013. It's called something like, does the message diffuse faster than the messenger? Something along that lines. And they actually go through how these two, you're measuring different timescales. The FCS and the FRAP are measuring different things. One is measuring your short timescale dynamic movement. One is measuring the large scale dynamics, taking into effect all the binding and unbinding and interactions. And the two are not in competition. The two are actually mutually supporting each other. And so, in fact, when we, with our modern FCS measurements, we actually get, in the FCS, a slow timescale that matches the FRAP, which makes sense, because we're picking up the two of them. And if we then calculate from this, what is our effective diffusion constant, which is essentially the fraction of molecules which are in the fast and the fraction of molecules which are in the slow, you get something around 3 to 5 microns squared per second. If you compare that with the tandem reporter I showed you yesterday, when we do the fitting with the tandem reporter, we get a value of 3 to 5 microns squared per second, because the tandem reporter, this 2 fluorophore system, is measuring the long time scales. And so, in reality, if you think of bicoid as a single object doing one thing only, then this is the effective diffusion of bicoid. In reality, a pool of bicoid is moving damn fast. There is a pool of bicoid moving damn slow. Cumulatively, they have the effect of this diffusion. And if you have a diffusion constant of around 3 to 5, then in fact, the root mean squared distance is on the order of 300, I think, or 400 microns, which is the order that you need.
Wait, you get the square root of it?
Yeah, so it's well, this is 10 times bigger. So it's a factor of 3.3. So it's 150 or 200. But then, of course, I have not put in the multiple. So this should be multiplied by probably 4. It depends whether you think it's 1, 2, or 3D. So I think you get 300 or 400 microns when I did the numbers. Okay, so this is a good point. But
don't you think that this diffusion within the nucleus, it matters if you're looking at one thing contaminated or something?
Yeah, sure.
But this with FCS, you just cannot discriminate. You are just right?
Yeah, so you can, not in this system. I've actually done FCS in Pombi, Pigeon East, where we could actually see clustering. But there, the FCS was much cleaner because it's a much easier system to image. It depends how much. The problem with clustering is kind of a second order effect, and your noise is very high. So it's very hard to see in the FCS. But in principle, you can see it. It would come into these. Okay, let me rephrase it. Things like clustering would add another time scale. If you could fit that time scale and use the correct model, you could get a clustering dynamics. But the point is whether you can reliably extract the clustering timescale from the FCS curves. And I suspect you can't, because I suspect it gets merged into the binding one. Yeah.
Perhaps this is a stupid question, but is the fast diffusion of eco compatible with its resonance time? So is it moving too fast?
No, in this case, it's not moving too fast. It's actually an interesting question about search strategies or how you find locations. And in fact, one thing that FCS can distinguish is between one-dimensional random and three-dimensional random. So the shape of G of Tor does depend on your dimension. And so there are people who have done FCS on 2D surfaces versus, and so on membrane. So Torsten actually does a lot of work on lipid bilayers. And there you have to use the 2D fitting model. And in fact, in STS, you can do it with 1D on DNA searching. And so, in fact, Bill is by far more of an expert than me on this, but you can have a combination of both fast 3D strategy, where you basically want to move around to different parts of the DNA, and then you want to have 1D searching, where you diffuse on the DNA, but that would be much slower, and then you hopefully identify a point, and then you come off, do fast 3D searching, find a new place to bind, and then you move around searching for a so people have actually looked at this in some beautiful papers from Bill Bialek and others from about 10, 15 years ago, but I'm not an expert, so I don't want to go there. Okay, so I've just spent, okay, go on in, sure.
So does the diffusion coefficient that you get from FCS not depend on the elimination volume?
It does, totally. And so if you change the volume, then you obviously change, you want it to be ideally as small as possible, again, because you want low number. If you make it bigger, then you actually then reduce your fluctuations, but you kind of get some advantages. There's actually something called, is it called the area law? So what you can do is when you do your FCS, you can actually change the system that you measure over. And the correlations, the timescales should actually scale with the area. And it's a good way of checking if your FCS is working or not. So people do this, and it's actually a good control. And so, yeah, so in this formula for G of T, you have to know what the shape of your PSF is, or the area that you're imaging, because it can be bigger. And so that actually comes into that. Okay. I spent an hour on this, but I think it's important because this is kind of the essential elements. I hope that essentially both are very good, both can be very informative. The key thing to really realize is you are measuring timescales, which is like a dynamic mode of the system, all right? That's what you are measuring. You then got to apply a model to interpret that into a parameter, a diffusion constant, a binding rate, a bound fraction. That's where trouble comes if you're not careful, all right? But I hope this is much clearer. Okay, so I'm not going to talk too much about stuff today. I think what I will do today then is, so tomorrow there will be a quiz on this, because why not? You can always go and look up. I think what I'm going to do today is then, I guess I can ask, do people want to hear more about morphogen gradients, in particular how opposing gradients can have a role, or would you rather me get onto like the readout and the feedback and so on? Okay, I hear more morphogen, so they win. Okay, so we'll talk about that for the rest of the day. I'm just going to rub off the board. If you want, I don't know. So the videos are made available, but not quickly, is it? I don't know. Okay. Yes. If you want to take a picture of the board, this is your chance. So, what I want to do then in the last 20 minutes is talk about two different things. The first is that, as ever, yesterday I did the usual, most of what I talked about, even though I criticized it, and I still did it myself, is that we mostly talked about the steady state solution to how we make a morphogen. So, write up our equation again. Your favorite equation, probably by now, or probably not. So, a lot of what we described, for example, the solution in steady state is this exponential. But of course, that doesn't have to be the case, right? So the question that some people ask is, well, okay, hang on, what happens if, can the system interpret prior to steady state, right? And some evidence for this came from the fact that in the early Drosophila embryo, that transcripts for target genes of bicoid were observed in sort of nuclear cycle 9, 10, 11, earlier than, so what Mooney was talking about was mainly 13 and 14. And so this was in the stage where the bicoid gradient was still forming, and so they're saying, there's what we call pre-steady state decoding. And so the idea here is if you plot rho, then of course what it does is it looks like this. And eventually you reach the steady state, right? So as time increases, your gradient increases. And if you consider the classic threshold model, so you've got a rho t. So then you've got to read off some threshold position. You can ask, well, is it possible to read off accurately a position in pre-steady state? Because the gradient is changing through this point. And it's a nice paper from Bergman. This is in PLOS Biology. 2007, where they argued that, in fact, you can do this. So if the dynamics of bicoid, they actually use a diffusion constant of around 3 or 4, is reasonable, then in fact, it can be advantageous to interpret the gradient whilst the concentration is low. And why is that? Well, one of their arguments was, in fact, that fluctuations in the production rate and so on are actually not so important earlier on. They're only really more important later, where these large-scale differences can have an effect. And so it turns out that you can solve this analytically. For those that want a clue, you might want to try looking up the error function. And so I don't advise, well, it's fun to do. It's a linear equation. You basically take Fourier transforms, and it's pretty straightforward, but it's beyond, I think, the scope of what we want to do here. But the point is you can actually get an analytical form for rho of x of t, and you can then do the same trick as yesterday. So then you can still do now delta x, but delta x is now a function of x and time, and so that's the fluctuations at that time over rho prime. And part of the point that they realize is that the fluctuations can be smaller than in the steady state if you do it in pre-steady state, and so they can be advantageous to interpret. Another thing that people had realized, and not in bicoid, you'll be pleased to know that we do actually think of other morphogens occasionally, is one in the worst named morphogen around, which is sonic hedgehog. There's quite a funny story behind this. It was, hedgehog was discovered in Drosophila. As ever, Drosophila has the world's, I'm in them, I don't know where Mooney stands on this, so I should be careful, but I hate Drosophila naming conventions. I think it's extraordinarily unscientific and unhelpful. So hedgehog is named hedgehog because you kind of have a spiky phenotype in the bristle hairs. And so it's called hedgehog. Then that was, I think hedgehog was discovered in the late 70s, early 80s. I can't remember, but I know that Phil Ingham worked on it a lot. And then Phil, they got interested in, well, is there an equivalent in vertebrates? And so he, along with some others, started investigating. And in fact, between 3 labs, they were all together on an HFSP grant, all discovered sonic hedgehog. That was not its name in Phil's lab. It was the name in one of the other labs. I think a postdoc apparently just coined it this because, of course, it was hedgehog in flies, and so he probably had for those of you who are old enough, in the 90s, Sonic Hedgehog was the game to play, so if you go to Phil's original Nature paper on this from about 93, 94, they do not call it Sonic Hedgehog, but that was unfortunately the name that stuck. And Phil was telling me he actually was giving a talk at so it turns out this is a really important oncogene, for example. It's a growth factor. He was giving a talk to doctors, like medical doctors, and his first question at the end of the talk is, why did you give it such a stupid name? I've got to go to these parents have just had a kid who's severely deformed because he has a mutation in Sonic Hedgehog. So I think it's a good example of where our naming conventions could maybe be a little bit more, has some real world effect. But yeah, I'm very much in the anti-naming convention, but that's the way it is. Anyway, so Sonic Hedgehog, to get back to the point of what I was talking about, is a very interesting morphogen. It's a very weird morphogen. It doesn't really diffuse properly. It needs to have binding partners to diffuse. It's a lipid, I think, and it has cholesterol binding sites, and it's all sorts of weird stuff. But there was a paper from the early 2000s, I think it was, that actually said, well, if we look at the response to a system, we see patterning as the level of Sonic Hedgehog is going up, but we see nothing when it sort of levels out. So the response was not to the total level, but it was a response to the change in the signal. So they managed to use different genetic perturbations to affect the levels of Sonic, and they basically saw that the responses were dependent on the time of the change in the signal, not the absolute value. And there's been more recent work by Eric Sigier and Iron Warmflash, Warmflash was in Eric's lab, but he's now got his own lab, where they use a TGF, they show similar things with TGF-beta, using a more synthetic system, which is more controllable, where they can pump in and out at specific times and see responses. And so the key thing here is that, in fact, it turns out that it may not be that the system is responding to the concentration, but it is responding to the change in the concentration. And there's a few reasons why that may make sense, because actually controlling absolute levels is really hard. You think about all the parameters that have to go into regulating exact protein numbers. That's seriously non-trivial. But if you kind of want to just, okay, I'm going to change from state A to B, that change, and that's what I'm measuring, that's probably a more reliable, well, not probably, it can be a viable signal to read out. And so if you actually imagine that we now plot the concentration along at a specific position, so we now plot the concentration along a specific position, but instead of plotting rho, we plot the rate of change. Then in that time, it actually looks like this, right? And so you could imagine that actually I have a threshold where I respond to a change in the signal rather than a specific value. And that way, I can, for example, define a precise time window for interpretation. And so this comes back to what Mooney was talking about this morning, is you have precise readout of genes at precise times. How do you regulate that? In the classic morphogen model, you just read out a concentration. How do you know when to read out a concentration? And in fact, tomorrow you'll be pleased, I'm not going to scribble on the blackboard, I'll use slides. And I'll actually show you how Bicoid, when it is actually reading out specific, when Bicoid is being read out for specific targets. But so here you can actually now define like a switch response. But of course one can ask, well, how on earth do you actually read out a response? And it turns out to be surprisingly simple. So we have our morphogen. Let's say it upregulates factor X. This upregulates factor Y, which inhibits factor X, and this can degrade. So if we write down the dynamic equations for this, and I'm just going to ignore diffusion. So we assume that the production is dependent upon something times how much morphogen we have. Now I'm choosing parameters simply because they make the maths easy, and actually, well, they give a specific solution. But so my x is it degrades at some rate, and it can be inhibited by y. So y basically decreases it. And then we can, obviously there's one for dy by dt. Right? So y is produced by x, and then x can degrade and it can be inhibited by y. And so if we differentiate this again, I'm just going to use the dot notation because I don't want to write the a to x. So now we can have beta rho dot minus 2 beta gamma x dot minus gamma y dot. But y dot is equal to beta x, so let's, so this becomes minus gamma beta x. So for the physicists in the audience, of course, I hope that this is standard stuff. So of course we can now plus 2 beta gamma x dot plus gamma beta x is equal to beta rho dot. And you can hopefully see that the solution to this is equal to something of some exponentials, which I can't really be bothered to write out, plus. And so if you choose your beta and your gamma to give you the right timescales, essentially the time-dependent parts here are small compared to the this rho dot term, and so therefore, effectively, your level of x is dependent upon the change in the morphogen. And so this is a very nice, simple model whereby you can actually now interpret temporal information. And we can also then go to Leslie's lecture and you say, well, okay, that's all well and good, but if you're measuring a derivative, you essentially have to make two measurements to measure the derivative, all right? And you can actually, there's been models proposed for measuring out the spatial derivative as well. You always have to measure 2 points and you have to compare them. So yesterday we talked about the measurement error, right? And that was measuring 1, but now we've got to measure 2. So essentially your error is increased. But I'm not going to go through it in details, but and me, I guess I put my name up, but so David Richards, who's at Exeter, and me wrote a paper in Biophysical Journal a few years ago, where we actually went through all these gradient models, so rho dot or d rho by dx, and asked, well, can you actually measure these out precisely? And it turns out that if you have time averaging on reasonable timescales, then actually you can. It looks like that in the Bicoid gradient, the timescales are too fast. You can't really do it for Bicoid, but the action of sonic hedgehog is on the order of hours. And so on those longer timescales, it actually looks like this is a plausible mechanism. Yeah. Sorry?
What are the two genes?
I actually, so this model is taken from Eric when their system where they looked at TGF beta. So actually I can't remember, you'll have to check. If you either go to, there's a beautiful paper with, Aaron is the first author and Eric is the last, where they actually developed the TGF beta, so not for this, but they did it for, but then for sonic hedgehog, it's actually, we applied it to the neural padding of the nerve cord. And I can't remember what X and Y would be, to be honest. Well, yeah, but that's, so we're going to cover that tomorrow. So I will cover 3 gene networks tomorrow, we'll get there. But that was, we had the choice and people shouted Morphogen, so the Morphogen won. So this is showing that you can have temporal readout. There's just, so lazy answer, it's because when you write down now the wave equation, this is basically a wave equation, right? And if you now solve this, you get a common denominator. So if you do this as m's, you do the usual way of solving this. So I'm just going to call this a m plus a squared. This is squared of this. So this is just. So it's just so that it's a. I took it from Eric's paper. The point was just to show that you can make it that it's oscillate, that you can read out rho prime. By using this particular combination of parameters, then you make the solution to this equation, very straightforward. But that's kind of not the point. It just chose that to make things a bit simpler. But these can be whatever you want. But the key thing is that you need, essentially, there's some prefactors here and so on. You want the time scale of action of this to be much slower than the time scale of action for this, so that the system is responding to the rate of change. So you need to choose your alpha, beta, and gamma so that this term is dominating. So you can kind of see you want gamma to be small, and probably I think it's beta to be big, but I need to check. No, there isn't a physical interpretation. It's physicists. I want easy maths interpretation. Oh, I don't know. Yeah, that's a good question. Is that the question? Sorry. Yeah, that I don't know. I just assumed it's because I only wanted. I don't know. I didn't want too many parameters. I could change this. Yeah, this could be whatever. Yeah, it's just simply I didn't want to make it too complicated. Yeah.
One comment. There's another strategy.
There is, and I hope you attend tomorrow, and you will hear about it. So I will talk about incoherent feed-forward loop tomorrow, if you don't mind, because I want to talk about network motifs, but I want to do that properly. I don't want to rush into it now, but you're absolutely right. You can generate a pulse generator with an incoherent feed-forward loop.
So I think this is regarding the pre-steady state interpretation. But doesn't that, like as other Lander points out in one of the videos, doesn't that come at a cost that now you have a specific time at which you need to start interpreting?
Absolutely, you do. And I will show you tomorrow evidence that this isn't true, right? Okay, no, let me rephrase it. That's not true. There is pre-steady state readout of the Bicoid gradient. There are transcripts of hunchback, of giant, of Krupel produced in the early embryo. They are completely and utterly irrelevant to successful patterning of the embryo. And so, in fact, I think the argument really is that these things happen, but they happen because stopping them happening would be really difficult, and I don't care that they happen, right? And I think this is a case where people have often overinterpreted stuff. So people have seen transcripts in cycle 11, so they go, there must be something important, regulation about cycle 11. There isn't, actually, in my opinion. It's simply that the system is primed to produce these transcripts. It mainly wants to do it in nuclear cycles 13 and 14, as Munio kind of was covering, but it happens earlier. And okay, I might as well talk about it. I'll finish on this. So my lab made, so how do you know if this is the case? So my lab made a bicoid with Cry2. So Cry2 is an optogenetic molecule. It dimerizes upon light activation. So if you shine 488 light on it, it clusters. And we made this line, and it was very nice. When you keep these flies in the dark, the embryos develop totally normally. We would do the nice Drosophila genetics trick where we could take our line, we could cross it to the bicoid mutant, so that the only copy of bicoid in our fly was this one with the Cry2. You keep them in the dark, they're totally happy. All the gap gene boundaries, so the hunchback Krupel, so on, are in the correct place. Basically what Munio was talking about, this all happens totally normally. You turn the light on and all hell breaks loose. And in particular, what we think is happening is that I don't know where I draw a graph actually. The bicoid is a transcription factor. It's meant to bind to DNA. So you've got binding sites. And what we think is happening is that the bicoid comes in. Bicoid is known to cooperatively bind. So you've got, say, 2 bicoids here. And under normal conditions, pol 2 or whatever comes along, tootles along, reads them out, you get transcription. But if you've got this Cry2 here, so in the dark, it's like this, it's all fine. But then if you activate with light, then this is, by the way, purely a model of what we think is happening. We think that the Cry2 essentially dimerizes and essentially conceals the bicoid from the transcriptional machinery. And so the details are kind of unimportant. What happens is it turns the embryo into a bicoid null. Embryo is unable to interpret any bicoid signaling. And so we can basically, by turning light on at specific times during the early development, tell you when bicoid is needed. And so we actually did that. So we went from nuclear cycle 1 to, I'll just skip some bits, 8. So we could say basically, okay, if we keep the embryo in the dark, okay, I got 5 minutes, I guess. If you keep the embryo in the dark for 5 minutes, actually, it's not far off. If you keep the embryo in the dark for about the first hour of development, then, and you if you keep it in the light for up to here, and then you turn the light off, so then it's just normal, the embryo is totally normal. So there are people that claim that because you can actually see hunchback transcripts in nuclear cycle 5 and Kruppel transcripts in nuclear cycle 5. They're just irrelevant. They're just because the machinery to make them is there. It's going to transcribe a few, but the numbers are so few that they're not important. If you keep the light on to nuclear cycle 11, then it's mostly okay. What you actually see is some shifting in the boundaries. And this actually comes back to the question I asked Munir yesterday, which is, bicoid seems to play a role in opening up chromatin for its later activation. And what we're doing here is that Eric Wieschaus has shown that role of bicoid in opening up the chromatin is, or I don't know if it's opening up, but it's playing some sort of role in epigenetically changing the landscape of binding, is this window. And so if we don't allow that to happen in this window, the embryo still patterns fine, it's just slightly, all the boundaries are not quite where they should be, but it copes. If we do this, we get basically a null mutant. And then we can do all sorts of combinations. We can do this, and look at all the different variations. And so we actually, in the lab, were able to directly test this model and say, well, yes, you can read off in nuclear cycle 11. Yes, there are transcripts there. No, it doesn't care. And so in fact, and this makes sense, because the point here is that in this part, the thing to remember is that the number of nuclei is 2 to the n. This is your machinery for making stuff, right? So essentially, after each division, you have twice as much machinery for making stuff. So you have far more transcripts are produced in the last 2 or 3 cycles than all the other cycles combined, just by simple exponentials, right? And so, yeah, there's some stuff going on here, but like my argument would simply be, it's too much effort to stop it happening. You need to have some sort of epigenetic mark that would inhibit the readout of bicoid for a certain amount of time that somehow magically knows when to stop being there and come off. Just screw that, just let numbers win. As you increase the number of nuclei, you increase your production power. So when you want to read out is when you're going to be reading out the most. And essentially that will overwhelm any earlier mistakes. And I would say that that's, to me, is the most plausible evidence. So optogenetics, so Munir's already alluded to it, is an extremely cool tool. We use it a lot in our lab to where you can temporally and spatially actually control morphogenesis. So we could do all sorts of cool things where we could take an embryo and we could illuminate half of the embryo and then get all sorts of weird stuff going on. And that's flashing at me now in red. And I think that's serious. So I will stop there. I haven't covered barely anything I wanted to, but I actually hope this was very useful. So tomorrow, as already, I like these, a lot of the questions have been good leading questions. So tomorrow we're going to cover networks. So how do you actually take this morphogen signal and interpret it? So we're going to talk about network, coherent feed-forward feedback, auto-regulation, and I'll talk about some specific examples. I was going to give it as I probably will give it as a board talk again, actually. I changed my mind. And yes, there will be another quiz. So the quiz will be on FCS and FRAP. All right? I think it's worth doing because it's worth testing. And finally, so who was the winner again? It was, so congratulations. So I hope he doesn't win again tomorrow. This is on you now to win. Okay, I guess I've overrun. So if you've got questions, just come and talk to me at lunch and I'll go from there.